Coagulase test. Purpose. The coagulase test is typically used to differentiate Staphylococcus aureus from other gram-positive Kochi. Principle. Staphylococcus aureus is an opportunistic pathogen that can be highly resistant to both the normal immune response and antimicrobial agents. Its resistance is due, in part, to the production of a coagulase enzyme. Coagulase works in conjunction with normal plasma components to form protective fibrin barriers around individual bacterial cells or groups of cells, shielding them from phagocytosis and other types of attack. Coagulase enzymes occur in two forms, bountcoagulase and free coagulase. Bountcoagulase, also called clumping factor, is attached to the bacterial cell wall and reacts directly with fibrinogen in plasma. The fibrinogen then precipitates, causing the cells to clump together in a visible mass. Free coagulase is an extracellular enzyme released from the cell that reacts with a plasma component called coagulase reacting factor CRF. The resulting reaction is similar to the conversion of prothrombine and fibrinogen in the normal clotting mechanism. Two forms of the coagulase test have been devised to detect the enzymes: 1. The tube test and 2. The slide test. The tube test detects the presence of either bound or free coagulase, while the slide test detects only bound coagulase. Both tests utilize rabbit plasma treated with anticoagulant to interrupt normal clotting mechanisms. The tube test.is performed by adding the test organism to rabbit plasma in a test tube. Coagulation of the plasma, including any thickening or formation of fibrin threads, within 24 hours indicates a positive reaction. Figure 7-24. The plasma is typically examined for clotting without shaking after about 4 hours, because it is possible for coagulation to take place early and revert to liquid within 24 hours. In the slide test, bacteria are transferred to a slide containing a small amount of plasma. Agglutination of the cells on the slide within 1 to 2 minutes indicates the presence of bound coagulase. Figure 7-25. Equivocal or negative slide test results are typically confirmed using the tube test. 7-25 Coagulase slide test. Emulsions of Staphylococcus epidermidis on the left hand, S. aureus on the right were prepared in sterile saline. Agglutination of the coagulase plasma is indicative of a positive result for bound coagulase.